I will talk a little bit about air pollution in Bosnia and Herzegovina, and also present some results from the research in the field of lichens and mosses. How do we assess the impact of human activities on natural ecosystems? As we can see, there are a lot of anthropogenic activities. Humans are using different industries, cars, activities on the field, doing some activities on the soil, using water, surface, groundwaters, and so on. What can we do regard to this? What can we become, which information using biota? What can biota tell about the environment, and what is the response to natural stress? So we can use species that tolerate a limited range of chemical, physical, and biological conditions, and we can use it to evaluate environmental quality. Still, we have a lot of modern, but also traditional or conventional technology methods or analytical chemistry methods that we use that are advanced methods, but we still are trying to turning our scientific research to use the biota to tell us the story about the world and to become the information about the pollutants, the level of pollutants, their fates, their chemical and physical properties. So we can use environmental monitoring. What is environmental monitoring? We use it as a system of successive observations of environmental elements in space and time. We can say of pollutants that you heard about them in the morning. I will present because our research is more orientated to inorganics pollutants, but we are also doing, you will see, a lot of organic pollutants with our colleagues in some collaborations. Environmental monitoring has some goals, and this is to collect data of quantitative and qualitative nature on the presence and distribution of pollutants in the environment, monitoring of emissions and emissions in the environment. So the goal is also to collect data about the source of pollution and their distribution, as well as to become the information about the transport of pollutants and the determination of their concentration or levels at certain measuring points. We can achieve it by using environmental monitoring. In the literature, we can find different types of definitions of environmental monitoring, the types of them. Here I have written just a few of them, like air monitoring, remote monitoring, waste monitoring, monitoring of waters and soil, but also Yasmina told us in the morning something about this. I only want to mention maybe these pictures. The picture about air monitoring is a picture of the city of Sarajevo. Unfortunately, it looks very beautiful, but if we look more in detail to the picture, we can see that this is the autumn period in Sarajevo during 2020, and probably there is a higher level of pollutants in the air if we watch the picture. Monitoring of waters also and soils. This picture represents water in the Herzegovina part of Bosnia and Herzegovina, and it looks nice, but still there are needed some monitoring to obtain data to see if the level of pollutants are according with the legislation that Gordana has told us about. How do we conduct environmental monitoring? What is most important? actually to find the suitable location to do the selection of the sampling sites, to do and to choose the monitoring system, the monitors. We need a very detailed sampling plan and examination when doing environmental monitoring, and also it's very important to do the monitoring to choose the adequate and most efficiency method of detection of the pollutants. I will just briefly talk about all these terms because of the time that we have for the presentation. What is biomonitoring? As a part of the environmental monitoring, it implies the application of living organisms as bioindicators of change in the environment in space and time. The first time when it was used as a term bioindicator, it was in 1920. And Clemens denoted organisms that clearly indicate the ecological conditions of one habitat in the presence, their presence in the habitat. When we use monitoring, then we use living organisms at the level of ecosystems, communities, population, individuals, tissues, cells, and we monitor the state of the environment. What is very important to say that using biomonitoring, by using biomonitoring, we can have a direct assessment of the state of the ecosystem. Since I'm a chemist, when I first started to use mosses, lichens, but also trees, conifer needles for the assessment of metals at specific areas in specific times. I was a little bit confused about the terms because I'm a chemist of biomonitoring and bioindication, or bioindicators and biomonitors. So the terms of bioindication and biomonitors are interchangeable, and bioindicators actually qualitatively assess biotic responses to environmental stress, like if you have a specific lichens on some area, their presence can indicate poor air quality, or they have a specific response to very specific pollutants like sulfur dioxide or nitrogen oxides, the presence of particulate matter, and so on. Biomonitors quantitatively determine a response. The example is reduction in lichen chlorophyll content. That's the difference between these two terms. Because I want to present you some data about lichen and mosses models in Bosnia and Herzegovina, just for remembering that lichens are a symbiosis among cyanobacteria, algae, and fungi, and mosses, they belong to bryophytes, are frequently used as monitor in air contaminations. So they can be used as biomonitors and bioindicators. They are very powerful bioindicators on the grounds since they have no roots, and they acquire all their supplements from immediate introduction to the climate. Also, what is one advantages? Their high surface region to volume ratio further supports the theory of their use as bioindicator, or supports actually that they are able to capture pollutants from the air. As I said, since I'm a chemist, we are also in our group at the Department of Chemistry are doing the research in air quality and pollution by using different methods and examine different pollutants in the air. Especially, we are working in the field of a particular matter and the characterization of PM10. I didn't put any data about this because we had these lectures in the morning, but probably you now know the differences, what is on the left and right side on these quartz filter papers. What do you think? Which pollutant is collected on these filter papers, quartz filter papers? Yes, please. Yes, it's PM10. Actually, it's PM10. Thank you. As you can see, here are also some samplers that Yasmina described in the morning, and we actually have also a few of them at our faculty. Why is this important to point out? As I have said, in Bosnia and Herzegovina, a lot of cities lay in valleys. If they lay in valleys, then if you look in the case of temperature, if some very specific meteorological conditions occur, like temperature inversions, during the colder months, when it's autumn and winter, the pollutants get stuck to the ground, and then we unfortunately have very bad and poor air quality in our cities. And as you can see, they are taking these pictures are Sarajevo, actually, from different areas that are higher than the ground level. And they have been taken during the autumn period. If you look the picture, probably I can press here. If you see the picture here, it's a summer period, but still we can't, looking at the picture, not say that it's not polluted regarding the organics or so on. And I will show you one paper that we have done with our colleagues from IMI. Probably there you can find some data also about the organics. If you look to the picture here, what you can see on the picture, Sarajevo has a lot of small houses with a lot of chimneys, and unfortunately, the people are using a lot of wood and coal for the domestic heating as well as for cooking. Why I'm saying this? Because the potential source of some of the pollutants could be the heating of the houses and also cooking. And here, as I said, I didn't put any data about the metals that we have obtained in PM10 or dust samples, because we also conduct dust samples. But what have we also done? We have data about the total concentration of metals, especially cadmium, nickel, lead. As Gordana told us, they are in the European directive regarding air quality. So we have done the analysis of these metals, conducted the data for 10 years. And unfortunately, also we have done the health risk assessment, and the obtained data have revealed that the people from Sarajevo are exposed to higher concentration of cadmium in Sarajevo. Also, we have done the research in the field of the bioaccessibility of metals, also in PM10 samples. If you want to check what is happening with the sample when it gets to our body, and the sample has been conducted also in urban and rural areas of Sarajevo, and also the data on the results revealed that the cadmium, nickel, and lead concentrations, bioaccessible concentrations, are relatively high in PM10 samples. And as you can see on the right side, there is a paper published with the colleagues from the institute here in Zagreb. We have collected we have collected also PM10 samples, and they have analyzed the organics, and they have been compared to the results in Zagreb, and also, unfortunately, concentration in organics in PM10 samples. Here also we have done biomonitoring. There are papers that describe the using of lichens, Hypogymnia physodis, and also mosses, Hypnum cupressiforme. In different areas of Bosnia and Herzegovina, about more than 40 samples have been conducted and have been analyzed regarding the presence of heavy metals in them. And the results state that there is a very high naturality or alternation to middle naturality and alternation of these metals in these lichen samples. If someone also is interested in the results of also metals in PM10 samples, but the characterization was, or the determination was, using different analytical methods. We are using usually the atomic absorption spectrometry, the flame atomic or electrothermal atomization, but also one part have colleagues from IMI, and they have done the analysis by using inductively coupled plasma mass spectrometry. So if someone is interested, I can talk with you if you have more questions, or I can send you these papers if someone isn't interested. And here I have put only one slide about this PM10 concentration that you can see from 2010 to 2019. Unfortunately, the PM10 concentration is usually exceeded during the winter and autumn period in Sarajevo. The concentrations, sometimes they are even 5, 6, or 8 times higher than the EU reference value for PM10. What can be the reason for this? The air quality is mostly affected by emission of pollutants from industrial facilities, households, and traffic. As I have said in the beginning. And here, why did we try to use moss and lichens as well-known bioindicators to assess the levels of metals in them? Since we only are limited with a small number of instruments that we can use, or we can't bring them to all of the places to do the measurements, then we picked and are trying to find the most suitable mosses and lichens that we can use to assess the metal concentration and to become information about the air quality in Bosnia and Herzegovina. Because we can cover larger area than only one city or only a specific urban or specific rural areas. I put just one slide regarding the biological and health effects of pollutants, atmospheric pollutants. Here, as we can see, they undergo a lot of processes when we look to the acid deposition, or when we look to the gases pollutants, they come to the plants or to the lichens, because they are not plants. They can have some processes that lead to damage of them, and we can follow these processes by follow the concentration in soil, in the species material, or also do the correlation of these elements in samples collected by using reference standard methods. So this was our questions. What we can do, what we can use, mosses, lichens, which plants we can use, because we want to cover a wider area to become even quantitatively or the first data about inorganics in the air of Bosnia and Herzegovina. Is someone using in their research mosses, lichens, or other plants? Only animals like fish, probably. Fish, is someone using fishes for the determination of inorganics, organics? Okay. There are a lot of early published papers that suggest the various biological samples to be used to assess the state of the environment, such as soil, tree bark, lichens, mosses, and leaves. What we have found among biological samples that conifer needles and mosses are commonly used to detect atmospheric metal deposition in terrestrial ecosystems. And you can see some of the trees that we used for our research in different areas of Bosnia and Herzegovina. So the first research that we done was the biomonitoring of metal levels by using conifer needles and mosses. In one part in the western part of Bosnia and Herzegovina, it's Bosnia and Podrinje canton. We used conifer needles and mosses. What is specific for this area? This area is there are a lot of industries, especially military. There are military industries for obtaining different military products. What we have done, we have tried to determine the concentration of cadmium, chromium, copper, iron, manganese, nickel, lead, and zinc in these samples by using flame atomic absorption spectrometry. And I have presented some of the results, the content of these elements in these samples. As you can see, only chromium in mosses and conifer needles has not been detected by using the flame atomic absorption spectrometry. And all the other metals have been detected. Maybe what is important to say that the samples have been conducted from different areas for about more than 20 sample locations. They have been brought to the laboratory. Then they have been air dried. Not washed, because you can wash or either not wash the samples and use them in your research. And then we have prepared them by using acid digestion, using nitric acid and hydrogen peroxide on higher temperatures, and applied these solutions to the flame atomic absorption spectrometer. What is very important, what Yasmina also Gordana said, these methods, if we want to compare our results, it's really hard, because these methods are not standardized, and it's not so easy to obtain, to follow the protocol for good quality assurance and quality control. But even we can do spiking of the samples to have some information about the quality control of our methods. Sorry, I'm out of time. Sorry. Okay. What is the most important thing, that the obtained results suggest that mosses are suitable bioindicators for the monitoring of environmental pollution, and also, unfortunately, that the air in this area is relatively loaded with heavy metals. Also, just briefly, one other research was that we used lichen mosses and conifer needles in Sarajevo canton, near Sarajevo areas, and also these are the pictures of the samples. Also, what we have found that the lichens, mosses, and conifer needles are suitable for the determination of the investigated metals in the air of Sarajevo. You can also do the correlation analysis to obtain the potential sources, to have more information about the potential sources of these metals in the area of Sarajevo. Okay, I will skip this. What is our the biggest goal is that we need to have the zero pollution action plan to meet all the goals set there, and I think we can use biomonitors and bioindicators for the assessment of metals in specific areas during a specific period of time. This is one question for you and me and everyone in the world. Yes, bioindicators very helpful, straightforward, reproducible, cheap, effective, and what is more important, use tools that do not alter natural system chemistry after being utilized.